So I guess I'm one of the more ancient people at this meeting here, so perhaps I should start in the dim and distant past of the 90s, where a lot of protein domains got discovered and their structures were solved, and it became clear that proteins are highly modular. But some of those proteins were binding to small little peptide motifs in usually other proteins, and famous examples would be phosphotyrosine peptides bound by SH2 domains, or protein-rich peptides bound by SH3 domains. And no one was really collecting these into a database. So in 2001, I'd got a grant to start a database of these small motifs, and we started to collect them. And that's, a very mundane but important activity in biology, is getting things into databases. Biology is an information science. And we collected them, and then we realized that they were all clustering in these native disordered regions of proteins, which at that point weren't also really being investigated very much. And the first native disorder predictor from Dunker and his collaborators had been developed, but was commercialized. So we made our own ones once it became clear that there was a pressure point, and we had to start knowing the structural context of where our motifs were. And various other, I suppose, pressure points. We obviously wanted to be able to predict mathematically new motifs, and we've done that with moderate success, but not great success. And that's true of everybody, and it's a little bit disappointing that bioinformaticians are not good at finding these small motifs. So they're coming out of experimental science, primarily, the great majority of them, and then we're putting them into our database, and there's been a steady increase in the number of them being reported. We have over 200 different kinds and several thousand examples that we've annotated in our database, which is called ELM for eukaryotic linear motif. But then we run across other issues, and Norman Davey, who joined my group, he wanted to find viral examples of these motifs. And the background to that is that many of the cellular motifs actually had only been found after a viral example had been found by a virology group somewhere who were working on the interactions of viral proteins in those interactions. Obviously, the viruses have invaded cells, so their proteins are interacting with cellular proteins. And he kept finding that when he thought he found a new motif, it was already in the literature. So he got frustrated, but the sort of silver lining of this is that we collected lots of these examples, and we were so impressed by the viral strategies of taking over cells by using these short motifs that we wrote a nice little article with that collection in it, but pointing out really that this is the main mechanism that viruses use to take over cell regulation in cells. They have to do other things to cells. They need to replicate themselves and so on. But almost any part of the cell that you can think of, the virus provides its own motifs. They're often higher affinity and in higher number than the cellular ones, so they just jam the system. And famous examples of motifs first found in viruses would be ones which bind to the retinoblastoma protein. The retinoblastoma protein is named after a cancer type because what it does is to regulate cell cycle, and it's an inhibitor of progression of cell cycle. Now, DNA virus likes to get into S phase in the cell cycle phases, and then from that point it wants just to make lots of its own DNA. So it needs to push the cell into S phase and then stop it there. And it'll have a few different motif combinations that allow this to happen. And then the cell just produces viruses until it basically disintegrates. So these are examples for that story. Now, very recently, to come very much further forward, we've just published an article with Peter Tomper, who is obviously a major player in this meeting here. And it has become clear that these motifs are exceptionally abundant, and this says important things about the nature of the cell. So we have done some rough estimates, and our article is entitled "A Million Motifs for the Molecular Biologist," and the title is a play on some earlier articles. But By the time we add in hundreds of thousands of phosphorylation sites and many other post-translational modifications, we're fairly confident that we're going to get into the million further numbers of these short motifs, even though we only have 20,000 proteins in our proteome. So that's a manifestation of how complicated the cell is. And one of the problems that we've been having over the last, well, several decades is that as people have studied cell regulation and cell signaling, they've tended to publish results which oversimplify the system. So they may be able to state that, under the conditions of the study they've done, say P53, which regulates apoptotic gene expression, behaves in certain ways. But it's become clear that if you change the cell type, change other conditions, then everything you learn doesn't necessarily apply. And the other major problem is the amount of networking. So it's not really correct to think of pathways, except in the sense as that those are information transfer routes through a network. And it's certainly not correct to think in terms of kinase cascades, which is this very common metaphor that is used in describing signaling processes. Because at every step, there's negative feedback control that can be switched on. So you can have negative feedback control and you get lateral control. So the famous cascading pathways like the MAP kinase one are really, and they're presented as a cascade, it's extremely misleading. And therefore it's important for us to come and tell the research community just how complicated the cell really is, and that we've got past the point now where we should be oversimplifying things. It's not helping us to go forward. And in combination with some other technologies that are available now, such as the ability to engineer cell lines so that you can make a stably engineered stem cell from a mouse. From that stem cell, you can actually create new mice. You can create mice with something you've done. Might be as simple as put a fluorescent marker into the cell, but now you can track that protein in a mouse, and if the mouse develops, you know that any manipulation you've done to the protein has not had a dramatic effect on the cell regulation. So people can now start doing these sorts of experiments. They also, of course, can sequence whole transcriptomes, and one of the things that we've learned over the last couple of years was that the protein motifs that we work on are very often in alternatively spliced regions of proteins. So another of the review type articles that we've written is called Lost in Pre-Translation, because our motifs are being heavily alternatively spliced. And so whatever the function of the protein is, it's normally described in terms of the globular domains and not these short motifs. So this alternative splicing is modulating the behavior of isoforms of particular proteins. And that might affect where they go in the cell, and it might affect how stable they are. It's one class of the motifs that we like working on are known as degrons, because they are sites which target destruction of the protein. So when that flag is activated, which might be the activation of a system to destroy the protein, or it might be that site gets phosphorylated and becomes active, and that protein is removed and eliminated and so on. And we see isoforms which have different destruction potentials, so that they're differently stable under different conditions.